thing. I was, first of all, looking at the different things that you should have included in your tech report, right? So the technical report is a little bit different than just, like your full lab notebook, which contains both raw and analyzed data. It's not as formal as a full final paper, but it is not as informal as just a lab notebook either. So it should have your um, analyzed data, not the raw data in it, and it should have appropriate amount of information to understand what is being shown. So in your lab report, the technical report, what you should have had was the final curve that you were using for quantification, right? So you would have had an image like this. That's going to be one of your things. The one that you were going to use for quantification. So in my case, it's a polynomial because that's the highest R square with its equation and the R square value clearly visible. That would have been one of the figures. Now, many of you showed it, but you showed it after you showed your quantification, which, so when you're writing a technical report, you want to think about what story you're presenting or how you are describing your data, right? So you have used affinity chromatography and you've gotten all these fractions. And now you want to know if there's some protein in it. So one graph that I did not, or one table that I did not include in here was possibly like a 280 one if you wanted to use the 280 absorbance. And then you wanted to not only know if there's protein in there, but you also want to know how much protein is there. And so if I'm looking for how much protein is there, I need to first prepare a standard curve before I can do any calculation for my unknown quantification. So if you show me a table with protein quantifications first, and then you show me a standard curve, you can imagine like looking at it or reading through your reports, it doesn't quite make sense, right? Why are you showing me the standard curve after the fact? How is it related to this table before that you've shown? Where did you get these quantification from in table 1 if you did not have a standard curve? So you want to make sure that your story makes sense, right? You can't just start a book in the middle. You have to start by setting the stage, telling what the characters are, telling what they're going to be doing, right? So you have to make sure that that linearity of information is present in there. So that was one of the biggest things that people weren't really thinking about how, what they were presenting and what it was supposed to show and how to interpret that data because you weren't putting them together in the right way. So the first figure should have been for all of you, the standard curve, because that's where you were starting. After you give that little blurb about, now I take that back. You could have had a table with your A280s, right? Those 280 absorbances, which just tells you the amino acid content. So you could have had a sentence or two saying, after affinity chromatography, or after the affinity purification of rabbit serums, several fractions were collected for both at the washing step and at the elution step. The amino acid content in each was examined by spectrophotometric analysis at 280 nanometer. And then you could have had a table, in their table 1, there was progressively less amount of protein present or amino acid content in each of the fractions for our washes or the elutions, indicating that the highest amount of protein was present in elution fraction 1, for example. So you could have had something like that. But then you would still have to quantify it, right? So then you can say next, a standard curve was created to quantify the actual amount of protein present in each of the fractions. And then you would need to show the standard curve. And you would say, the standard curve through Bradford assay analysis showed the best trend with polynomial equation, R square equal 0.9943, and then in parentheses, figure 1, right? Because that's your figure. and then you would have the figure, and then you would have its title and caption. And then you would go to the next step, which would be to show your quantifications. Now, in your case, you would have just had a few of them. In my case, I had all of them. So I'm just showing you an example for that table where I have my samples listed, my absorbances, blank corrected absorbances, and then protein concentrations in milligram per milliliter using the equation that I had, the final numbers, right? And then lastly, you want to confirm that what you extracted or that what you purified was indeed rabbit IgG without any other impurities. And that would be your this figure. So I just took away my, right? I cropped out and I'm showing just my portion of the gel, not anybody else's. You guys were supposed to do the same where you crop out the marker and just your two samples, right? Your wash and the elution. That's it. And then this is not fully done. I have annotated it on the top where I tell what is in each of my lanes, right? Actually, I didn't yet completely do it. So let's finish doing that. This was my marker lane, right? This is my standard molecular weight marker. So molecular weight marker. And then not only that, I want to make sure that I label some of these because I know what they should be, right? So I want to label some of them so we can make sure that my size for the protein bands are the correct place. So, and I could do that two ways. Many people will do it next to them, or you can do it even on top of them. It's up to you. When you look at papers, you see it both ways. So, see. And I always start with the top because those bands are going to be consistent. The bottom ones sometimes may have run through the gel or maybe very faint and not as visible. The top ones can be seen very clearly, and so it's much easier, right, to figure those out. So this one is 110. This 80. And I'm gonna, center them in a second. 60. And I don't want to necessarily do it on all of them. I'm just showing you how you can. It's more important that you do it on the main ones that are needed around the bands that you have, right? So this one is 50. Sorry. Oh, 30. One is 20. Okay, so once I have them, I can align them. I can group them, right? Oh, I left one. Let's put them all ungroup. Align, center, so they're all nice and clean. Group, and now you see, sorry, that I have created a marker lane that's annotated that you can clearly see, right? And you know what it looks like. In addition to that, there's still something more we can do to this gel to make it even better. I already had cropped quite a bit of it, but frankly, I can crop a little bit more because there's a lot of empty space down here that I don't need. So I could technically just, Drop it even further. There's a strange band here if I wanted to annotate it. And there we go. Now, the two heavy and the light chains, right? This heavy and the light chain, I would go ahead and label, annotate those as well. Really. This is gonna be my light, heavy chain, light chain, IGG, so whatever you want to write there. Okay. And then you can very clearly see those as well. Okay. So this would be a properly annotated, properly prepared gel that you can now make sense out of, and now you can explain in your results section and also show as a figure. This would be the figure, okay? So a lot of you just had like the full gels there with multiple groups data in there and not really explaining anything clearly about them. So that was something that I thought was lacking in many of your reports. Again, remember, the full rabbit serum contains lots of things, right? And you have the immunoglobulins, but you also have albumin, which is right around 60. You have other proteins that are right around there. So you have a lot of these. This big blob contains many, many, many proteins. And you have then other bands as well, both on top and the bottom. The wash through and the flow through will contain everything but your IgGs, or should contain everything but your IgGs. And in here, you can see the light chains are not there at all. And if you look here, this bottom band, which coincides with the heavy chain, right? This bottom area, that's no longer there either. That indicates that your beads, the protein AG bound beads, the affinity chromatography beads, work really well to bind the heavy and the light chain, the IgGs from the rabbit serum, so that your flow through was clean of them. And then when you look at the elution fractions, you can see that there's very little contamination with other. There's a little bit in the first elution fraction. I can see some higher bands, but a little tiny bit, but not a lot. Majority of what I'm getting is pure IgG, and it's clean of other contaminating proteins. Again, meaning that through our wash steps, we were able to remove anything that was not bound, anything that should not have been bound to the A protein AG beads or that had been just floating around. And what we are getting here is pure IgG, which is what we went out to do, was to purify the rabbit IgG fraction. Does that make sense, guys? A little bit?
Yes, it makes sense.
Okay. So this was something that you want to be careful with in the next one is one, the flow of your discussions, right? Flow of your technical reports. You also want to be mindful of how you are formatting your figures. Do they make sense coming together? And what are they talking about? So actually this time what I'm going to have you do is do a little bit different, where instead of having a single panel, you do what we call a multi-panel figure, where you're going to combine the data that is related into a single figure and then talk about it, hoping that that's going to help you see how things are connected to each other. And so you will end up with two, because of the data that we have, you will have two different figures that you will show. Okay, so this much for this. Now let's go through the actual assignment for the tech report and kind of, I'm not going to do the entire thing, obviously, but I want to do a little bit of it with you together to so you can see how it should have been written up. So, I went back and I looked at the instruction and I made sure that the things that I'm taking points off were, in fact, very clearly listed there. Um, a lot of you missed out on the results section, right? Uh, because you did not either write your titles and captions for the figures, or you did not describe them in the text, right? Um, so right here, it does talk about that the results section is where you're presenting your data. You're not interpreting it, you're just presenting it. You're not drawing any conclusions from it yet. That each figure and table should be incorporated into the text at the end of the paragraph where it is cited. That sentence alone should tell you and remind you, and many of you I've had at Mosel, so you know this. We have done this before, that you write, there's a narrative results text that you write about what you are presenting, your data in a paragraph form before you present the figure or table. And then below the figure or table, you are going to have its title and caption. That's a separate thing. And that was missing in many of yours. Some of you really just like this simple thing. So the first part was, let me open a Word document. Okay, so the first part was keywords, right? And many of you only wrote 3 keywords or no keywords, which was a little bit sad because, that's easy points. Why would you not write those? Let's see. Give me some keywords from the first module. What could we have done? What would be good keywords? We're going to do this together for a few minutes. IgG, okay. And, login IgG. Sure, I'll give you. Not the best one, but yeah, it's a decent one. Protein purification, or sure. What may have been better might have been, for example, instead of protein purification, might have been doing like antibody purification, because what we were doing was essentially how we purify antibodies, right? But yes, protein purification works. SDS page, sure. But even before that, what was the technique that we did first? I haven't heard that yet. We used a very specific technique over there. No, that's later. We will do protein assay before that. What technique did we use to purify our IgGs? Like rabbit serum, or just serum for that matter. Affinity chromatography, exactly. What was it in that column that helped you isolate IgGs from rabbit serum? What were those bindings? What was bound to the beads to help us purify the IgGs? Well, they were the matrix, but matrix alone is not gonna isolate IgGs. This was something bound to the matrix that ensured that we were gonna get rabbit IgGs to be isolated. What was it? So the matrix was in the form of beads that had those things bound to it. A particular type of agent bound to it. What was it? No, that's later, and that's for a very different function. No, it was an antigen for the IgGs, but what it was protein AG beads, right? Protein A and G, bacterial proteins, right? Remember we talked about that? How protein A had a high affinity for the rabbit IgGs, which is why we had them in there. So protein AG, right? Those were specific bacterial proteins that were needed for the affinity chromatography to work. Sure, we did STS stage. Beta mercaptoethanol could be, in a way, a keyword, by the way, if you wanted to use one. Use it as that because we were looking at the effect of proteins run on STS page with or without beta mercaptoethanol, right? And what is the beta function of beta mercaptoethanol? What was the function of beta mercaptoethanol? What? Seriously, control. Why do we add beta mercaptoethanol to our stuff, to our samples?
Is it to reduce the disulfide bonds?
Yes, it's to reduce the disulfide bonds and to linearize our proteins and to separate the heavy chain and the light chains from each other, remember? So they're not combined together into a big complex, but rather separated, and you can see those two individual bands. That's the function of beta mercaptoethanol. Okay, any other keywords we could use? Actually, even before that, somebody said protein assay. Protein assays can be 1,000 different things. That's not necessarily the best one to use. A better one would be to look for, we were doing quantification of proteins, right? So you could say protein quantification. And you could talk about the exact assay that we used for protein quantification, which was which one? What was the name of the protein assay we used for the quantification?
The Bradford.
Exactly, Bradford assay, right? And then again, we stained our gel, right? So we could say protein staining, and there was a particular stain we used for our, protein stain. That would be it, imperial blue stain, right? Which is based on Kumasi. Bradford is also based on Kumasi. So you could say Kumasi blue staining or Kumasi blue, because there are two different methods you're using to quantify proteins or to visualize proteins that are both based on Kumasi blue assay or Kumasi blue reagent. So you see how there were a lot of different keywords that could have been used for our particular assay here. Next, you had to do an overview. Now, overview is not a protocol, okay? That's something we want to remember. Overview, and let's go back and look at what it tells us too. It does not have a method section. It does not have an intro, a proper introduction section. The overview is meant to just kind of give the big picture. What was the big purpose? What was the big question being asked through our experiments or answered through our experiments? What was our general experimental design and its rationale? Okay, so it's not to put 2-3 pages of information on what you guys did and how it was done. It's to tell us the general step, your logic behind each step and just telling us why you did what you did. 4 to 6 sentences, right? That's probably what you're looking for. So you would have a sentence, obviously, on, rabbit IgGs being present as one of the main components in rabbit serum, and that you can isolate them through affinity chromatography using the properties of protein AG, protein A and G, bacterial proteins bound to the matrix so that they can be bound to it. So you could have a sentence on that. So, you know, the goal was to isolate rabbit IgG or, you know, IgG from rabbit serum to mimic the process, for example, of antibody purification, right? Through affinity chromatography using bacterial protein AG bound beads and matrix. Then you would say, okay, so the protein AG here, you know, and you can write a paragraph, a sentence about how protein A has high affinity for rabbit IgG, which would allow it to be purified. So that's the rationale for using protein AG bead bound matrix for affinity chromatography. Amino acid content in the wash and elution fractions was examined through A280 through spectrophotometry at A280 nanometer, because that's where amino acids light up, right? Followed by quantification of protein in each fraction against a standard curve derived through Bradford assay. Finally, IgG purification was confirmed through SDS page gel electrophoresis and through staining of SDS page gel. Maybe we will do 2 sentences here. Through SDS gel and IgG was confirmed through SDS page electrophoresis, followed by staining with imperial protein. Okay. Right? So this will give you the basic steps that you will be taking and why you did that. So you isolated IgGs through affinity chromatography. You confirmed protein content by 280. You quantified proteins through standard curve and Bradford assay, and you confirmed that what you had was indeed IgGs in your fractions by gel electrophoresis and staining. Do you see that? So just 4 sentences, that's it. And then you would move on to the next step, which would be the results. And again, I'm not going to write all of them. I'm just going to start off with the first part, and then we will move on to the next part. So in here, you would then write a sentence about what it is that you're going to be, or 2 maybe, about what it is, you know, that you were going to do. So you would say after affinity chromatography, you know, several wash and elution fractions were obtained from affinity through affinity chromatography of rabbit serum. These were to quantify protein in each of these fractions. A standard curve with BSA with protein dilutions ranging from, I'm just making up milligram per mil to 2 mg per mil, was prepared using Bradford assay. The standard curve was found to have a polynomial friend function with R square equals whatever, right? 0.997. I'm just making up a number. And then you would say figure 1. The equation, and then you would show your figure. Just a few sentences. I'm not asking you to write a million sentences for each thing, but 3 to 5 sentences for each figure so that we know what was done, what it shows, does it make sense, right? And then Control V, there's your figure. And then underneath it, you would have figure 1. What should be its title? Tell me a title for this figure. Are you guys there? Look to be there.
Yes.
Nobody's talking or breathing. I'm the one doing the assignment. So come on, tell me, what should I write as a title for this figure?
So we just mentioned the average absorbance at the 595 nanomoles.
So that's not nanomoles, by the way, right? This is the wavelength. That's nanometers. No, so your title should always be like the main conclusion that you can draw from it. And then in front of it, you would write your description or caption that can tell that it's the average from n equals 2 or n equals 3, however many you have, right? So for example, here I can say standard curve with our BSA, standard curve derived from Bradford shows polynomial friend function. And then in front of it, I can say, write down that BSA dilutions ranging from 0.25 to 2 mg per mil plotted against average absorbance at 595 nanometer, right? N equals 2 because you had 2 people that were doing the work together, right? Examined through Bradford assay. So just a sentence where I can see how many samples you had and what range you had, and that's it. Now, in MoCell, we've talked about how we can make this bold. It doesn't always, different publication papers, different journals will have different rules. Sometimes it's italicized, sometimes it's bold, sometimes it's nothing. It's the same as the rest of the text, but it is clearly different than the caption. So you have a title and then the caption. The rest of your stuff should be double spaced so you can clearly see when there is a caption, right? Otherwise, you can't even tell. You don't actually need this title there. Smaller, maybe there. That maybe over there. I know it's not the prettiest graph right now because I haven't formatted it properly, but it's all good. But I'm still making it look like this. Right, and then you would move on to your next section, which is to now show the table. And now when you're going to talk about the table, you're going to be, the equation from this, you have to make a connection with figure 1. You can't just, everything is not in isolation. You want to talk about how that first part is connected to the second. Standard curve was used to quantify the amount of protein in each fraction obtained from the affinity chromatography. Highest amount of protein was present in the flow through. Table 1. In addition, elution fraction 1 was found to have the highest level of protein in the three, each elution, or rather, in addition, each elution fraction had less protein than the one before, something like that. Table 1, something like that, right? So again, I would write another sentence in there about how much protein was there in the wash as well, but just to give you an idea, right? And then you would have your table, Control C. And tables, title and caption go above the table, right? So you would write it on top of the table in this case. And that one, so table 1, protein quantification, and fraction obtained after affinity chromatography. It does. I just read you that sentence, man. Abdul, I read that sentence first. I told you, I confirmed that it indeed said that, because we always say that. So right here, the data should be presented as clearly and concisely as possible in the text and presented in tables and graphs as appropriate. Each figure and table should be incorporated into the text at the end of the paragraph where it is cited. That's why I'm going through this. Why do you think I'm just spending an hour going through the assignment that you guys just turned in? This is exactly why. I want to make sure that everyone is clear on how it is done for the next time. Yes, I agree the instructions were a little bit differently written than I've used the exact same instructions for this class, because this is the class that this is the first time I am personally teaching this lab. These are the instructions that have been this way for many, many, many years, every year, same way. My instructions in MoCell are a lot more detailed because you guys are learning this stuff for the very, very first time. But a lot of you have taken me for MoCell and you know how these things go. Plus, it is in the instructions. It is definitely written in there that you had a results descriptive text and you had the actual figure as well. And I will also tell you another place where I did describe this as well. If you go back to the instructions given to you guys in that Zoom recording, I went over and checked in there as well. I do talk about that you needed to write a paragraph about it before you gave the figure and then the figure title and caption. Anyway, so yeah, so you will write the table once, title and caption, and then move on to the third part, which is to write about, obviously, then the next thing, which is going to be, oh, come on, go down. There. Which is going to be our final confirmation. So finally, to confirm that our illusion fractions contain that the illusion fractions, that the illusion fractions contain purified IgGs from Rabbit's serum. SDS page analysis was performed, followed by staining of the gel with imperial blue stain. Rabbit serum had several bands all across the lane, indicating lots of proteins present in the sample. Flow through and wash maintained most of these ending patterns, with the exception of missing the bands associated with high and or heavy and light, IgGs, flow through and wash fractions. Elution fractions had clean bands only for heavy and light IgGs, indicating we were the pure quality of the sample after affinity chromatography. Samples were treated, were beta or capital ethanol was added to the samples and heated before analysis with SDS pH, followed by staining of the cells with this. Only for heavy and now you get a pure quality. And here you would have figure 3. And now here is something that you could do. You could label these lanes 1 through 6, or you could write how I did, right? Silhouette 1 through 3. Silhouette or silhouette 1 through 3. Silhouette 1 through 3. Figure 3. Flow through and washing. And that's it. Okay. So this would be something that you could do here. And then you would have your actual figure. There is something missing in there in a second, actually. Right here, you want to also mention the size of the bands that you're looking at. Why isn't it going down? Why aren't you going down, man? heavy approximately 5 kilodalton and light and then you would have its title and caption under or figure 2, it's not figure 3. It's the second figure. Okay. So that's how you would do that and write now, in front of it, write the information about that. And then you move on to the discussion. In the discussion, you do not want to go back and talk about table 1 and figure 1A and figure 1B. You are going to talk about the data as a whole and talk about whether or not you were able to accomplish the goals that you went out to do. Okay, so let's go back and again, look at the discussion. This is where you interpret the data, right? We are not restating the data. We are not talking about the data again. We've already written those paragraphs talking about the data. We are talking about whether it supports our hypothesis, if we had a hypothesis, or if were we able to accomplish the goals that we set out to accomplish. You can explain the results if they are different than expected. You can talk about things that didn't work or worked. You can talk about changes in the future. You can talk about comparisons to published results. All those things, that's where those discussions are going to happen, right? So in here, I'm not going to write all of those, but for example, in here, you would, first of all, obviously talk about our results, no bold. The results indicate that affinity chromatography with using protein AGVs is a good method or is a, great method for isolation of rabbit IgGs from serum based on the pure nature of IgG in the SDS page stained gel, in the stained gel or even just in the stained gel in the elution fractions on the stained gel. And then you can talk about the quantification may not be very accurate because the standard curve was not very precise based on an R squared that was below, 0.95. In the future, we would want to use a better made standard curve for quantification purposes. You can talk about that if your samples were not run very cleanly, the gel did not show clear bands or did not show clean bands, possibly because they were not run appropriately or because there was mixing of samples from one lane to another. In the future, we would want to run these samples separately or rerun them using a new gel with lower voltage so they can be run more accurately and examined better. Those are the type of things that you can talk about. You had the two bands because you used beta mercaptoethanol to reduce the samples. We were able to detect both heavy and light chains separately. The sizes of these proteins were as expected. If yours weren't, you can talk about why they weren't as expected. What could have gone wrong? Maybe you heated it too soon and then it sat at room temperature for very long and came back together. Maybe you forgot to add beta mercaptoethanol and you only had one band instead of two. So those are the things that you would talk about here in the discussion, right? A couple of paragraphs and that's it. And that's where you would put in your references as well. Okay. Questions about this before we go on?
I have a question.
Yes.
So in the discussion portion, we're supposed to refer to an article.
Yes.
Yes. So would we make another paragraph in addition
Yes.
to making about?
So, you know, that paragraph can be incorporated in here. Like I said, you'll have a couple of paragraphs, right? So typically what you would do is stain gel. The sizes of bands were consistent with reduced, purified, you know, heavy and light shades. Similar results were seen in article XYZ, right? And then you write that reference in there. So that's how you do it. You have a sentence or two about that paper that you're trying to incorporate because you saw the similar sizes of the bands or you saw the similar method being used to isolate rabbit IgGs, whatever it is, and then you reference it in front of it. Okay?
Okay, thank you.
Camera on, so please be in some place that you can have your camera on. Outside of that, you will be just taking the exam, and once you're done, you are free to leave. Okay?
Sweet.
Okay, guys. So have a good spring break, and I will see you next week. Well, actually, I say that, but at the same time, I am going to go ahead and stop the recording, and then